Happy day students! Welcome to San Academy Group of Schools, Pallikaranai, Grade 8 Science. Today we are going to discuss about Chapter 2 Microorganisms, Friend and Foe. Why do we call these microorganisms both as friends and foe? It's quite annoying, right? How can we call a particular substance as both friend as well as foe? Fine. We are going to find out why it is called so in this chapter. Now, come on, let's get into the chapter. Many living organisms are present in soil, water and air around us. And some of these organisms are so small that we cannot see them with naked eye. And we need a magnifying instrument called microscope to see these extremely small organisms. And these extremely small organisms are called microorganisms, and they are present all around us, and they are so small that we cannot see them with the naked eye, and they are present in such a way that we can see them in air, water and soil. So, what are these microorganisms actually? They are very small organisms which cannot be seen with the naked eye and can be seen only with the help of a visual aid called microscope. And these microorganisms may be unicellular or multicellular. What does unicellular organism means? Unicellular organisms are those organisms which are made up of single cell, whereas multicellular organisms are those organisms which are made of more cells, that is 2 plus cells. And these organisms may exist either single or alone or in groups or colonies. And as I said you, these microorganisms are present everywhere, and they are also found even inside our body and on the surface of our body. Let us do a small activity to confirm that microorganisms are present everywhere. So, you can also try this activity, whereas part 1 you can do it in your home, whereas for the part 2 definitely we need the help of a microscope. So the activity is very simple. Take a drop of water from the tap and place it in the center of your palm. And do you see any organisms in them? But we don't, right? But when you place the same drop of water on the slide and cover it with a cover slip, and when you're going to observe it under a microscope, you can see so many tiny organisms which are floating or moving swiftly. And these tiny organisms which are floating or moving swiftly are nothing but what? Microorganisms or microbes which are present in water. So, the definition of microorganism is organisms that are so small and that can be seen only through a microscope, that is, which can be seen only through a microscope, are called microorganisms or microbes. Fine. Where can we see all these microorganisms? That is, where are microbes generally found? They are found in any kind of environment. And these microorganisms are found practically everywhere in all types of habitats. Microorganisms are found in air, soil, water bodies like ponds, lakes, wells, rivers and seas. And these microbes can live and survive in almost all kind of environment like hot desert, polar regions, ice cold polar regions, even in salt water, and few organisms are also found at the hottest region of volcanic eruption, and they are also seen inside the human body. And what is making these organisms to live in any kind of environment? These microorganisms generally have a hard cyst around them, and these microorganisms remain inactive during the unfavorable condition inside the cyst, and when the condition becomes favorable, they become active and they start multiplying. And this is how microorganisms are found in any kind of environment, both in the both extreme environmental conditions. Types of microorganisms. Microorganisms are generally classified into 5 groups. How many groups? They are classified into 5 groups, and they are bacteria, singular bacterium; protozoa, singular protozoan; algae, singular alga; fungi, singular fungus. And the fifth type, or the fifth group, is very unique because it lies between the line of living and non-living things, and it is nothing but virus. Now, let us discuss in detail about bacteria. The singular form of bacteria is bacterium, and these bacteria are very small. These bacteria are actually very small, very what? Small in size, and they are single cell microorganisms which have cell walls but do not have an organized nucleus and other structures. And these bacteria are found in large number everywhere, in air, soil and water. Every surface around us, on our bodies and even inside our bodies. Bacteria are larger than virus but still very small. Unlike viruses, bacteria feed, move and respire as well as reproduce on their own. And there are mainly 3 groups of bacteria based on their shape. And the spherical shaped bacteria are called coccus, whereas rod-shaped bacteria are called bacillus, and the spiral-shaped bacteria are called spirochete or spirilla. And these spherical-shaped bacteria can either occur in pair or in chain, and when they occur in a form of a chain, it is called streptococci, and when they occur in pair, we call it as diplococci. Alga. Alga, the singular form, and algae is the plural form, and it is a plant-like microbe which has chlorophyll, and they contain chlorophyll, so they can produce their own food by photosynthesis just like plants. But however, they differ from plants because they do not have proper roots, stems and leaves. Some of the example of algae are Chlamydomonas, Spirogyra, Ulva, Ulthrix and Cladophora. Protozoa, and the singular form is protozoan. And these protozoa are a group of single-celled microorganisms which are classified as animals because they are animal-like microbes. Protozoa are animal-like just as algae are plant-like, and protozoa are found in ponds, lake, dirty water drains, river, seawater and damp soil. Some common examples of protozoa are amoeba, euglena, paramecium, etc. And few protozoa can either live on their own or they live in the form of parasites, that is depending upon the other living organisms, and they are also found inside the human body too. And exclusively the protozoa Plasmodium is the reason for the disease malaria, which occurs in human, and Plasmodium is commonly known as malarial parasites. Fungi. The singular form is fungus. And fungi are a large group of organisms which are also plant-like microbe, but whereas they lack chlorophyll and hence they do not perform photosynthesis. Some examples of fungi are yeast, molds such as bread mold, penicillium from which the antibiotic penicillin is extracted, aspergillus and mushrooms. And yeast are single-cell fungi, whereas mushrooms are multicellular fungi which are big in size, and hence fungi occurs both in multicellular as well as in unicellular form. And most of the fungi are saprophytes, which means these fungi mainly feed on dead things like remains of dead plants and animals for their food. And some fungi are also existing in the form of parasites, where they live on living things and causes diseases. Friendly microorganisms. Those microorganisms which are beneficial to us are called as friendly microorganisms, and these microorganisms are used in several industries such as food and beverage industries, medicine and vaccine production. Even in agriculture they play an important role, and they also play an important role in cleaning the environment. Let us discuss about the role of microbes in food and beverage industries. First, let us discuss about the role of microbe in making of curd and cheese. Milk is generally turned into curd by bacteria, and in order to make curd, a little pre-made curd is added to a warm milk and set aside for some time. And this curd contains several microorganisms including lactobacillus bacterium, and this lactobacilli bacteria promote the formation of curd from milk. And when a pre-made curd is added to warm milk, the lactobacilli present in the curd multiply in the milk, and it finally converts the milk into curd. And this happens as follows. That is, let me explain you how it happens actually. Milk contains a sugar called lactose, and lactobacilli converts this lactose sugar into lactic acid, and this lactic acid then converts the milk into curd. And so we can say now that microorganism utilized in making curd from milk is a bacterium, and the name of the bacterium is lactobacillus. And after curdling of milk only, it is possible for us to make cheese, and depending upon the type of cheese we require, different types of bacteria are used in order to prepare the cheese from curdled milk. Now let us discuss about the role of microbes in making of bread. Generally, the most commonly used microbe in making of bread is yeast, and because of which yeast is used in baking industries exclusively. When yeast is mixed in dough for making bread, the yeast reproduces rapidly and gives out carbon dioxide gas during respiration. The bubbles of carbon dioxide gas fill the dough, and as you can see in this picture, it increases its volume by raising the dough, and this makes the bread raise, and the holes in the bread are due to the bubbles of carbon dioxide given off during the baking process. This makes the bread light, soft and spongy. For same reason, yeast is also used in making cakes and pastries. Now, let us discuss about the role of microbes in idli and dosa batter. We all know that idli and dosa is the most famous South Indian tiffin item. And how are we making these batters? And the idli and dosa batter are mainly made when the dal and rice are grinded together, and when it is left aside for some time, they undergo fermentation process. And how is this fermentation happening? Due to the presence of certain bacteria in the batter, and after some time you can see the batter becoming sour, and now it is ready for making both idli and dosa. The same way, there is another familiar dish of Gujarat called the dokla, whereas even in this dokla the batter has to become sour, but here it is not happening by a natural method, that is not by the presence of bacteria which are present in nature in the batter. Here we add yeast, and by adding yeast we make the batter sour, and which is now ready for making the dish dokla. And now let us discuss about the role of microbes in making of alcohol and alcoholic drinks. As we all know, yeast, during the process of fermentation, it is breaking down the sugar into carbon dioxide and alcohol, and these alcohols are then used as alcohol as well as in order to make several alcoholic drinks. But the only difference between the alcoholic drinks like beer and wine is the difference comes mainly from the source of the sugar. In case of wine, the sugar present in the grapes is undergoing fermentation, whereas in case of beer, it is the germinated barley which is containing the sugar is undergoing the fermentation process, and it is resulting in the production of beer. Now let us discuss about the role of microbes in antibiotics. And what are these antibiotics? Antibiotics are medicines which are used exclusively to kill or inhibit the growth of the disease-causing microbes. And mostly these antibiotics are produced from bacteria and fungi. So, a medicine which exclusively stops the growth or kills the disease-causing microbe is called as dash antibiotic. And the source of antibiotic medicines are nothing but microorganisms, which are exclusively bacteria and fungi. Now let us discuss about Penicillin. Before discussing about penicillin, the most famous antibiotic, we should know about Alexander Fleming. Alexander Fleming is a scientist who discovered the antibiotic penicillin in the year of 1929. When he was working with a disease-causing pathogen, he observed that in the petri dish in which he was growing the disease-causing culture, he observed there were few spores which was green in color, and he also noticed surrounding those spores there were no growth of bacterium, and then those spores were identified as penicillium mold, and these penicillium mold were secreting a substance called penicillin, which was inhibiting the growth of the bacteria in their surrounding. And so penicillin comes from a species of mold called Penicillium. And apart from penicillin, few other examples for antibiotics are streptomycin, tetracycline and erythromycin. And one more parameter which we have to keep in our mind while using antibiotics is we have to use antibiotics wisely. What does it mean? It means that antibiotics can be used only when it is being prescribed by the doctors. And even when it is being prescribed by the doctors, we have to complete the complete course of antibiotics. And we should not take it either in overdose or than the amount, lesser amount than it is being prescribed. Because if we take antibiotics unnecessarily, it may kill even the healthy bacterium which are found inside our body. And always remember, antibiotics are not effective against flu, and common cold, because those are the diseases which are caused by viral infection, whereas antibiotics are effective only against bacterial infections. Microbes in vaccine making. Vaccines are generally used in order to protect the human from several diseases. And based on what principle these vaccines are working? Generally, when a person is affected by a disease-causing pathogen, the moment the pathogen is entering into the body, his immune system starts producing antibodies, and these antibodies fight against the disease-causing pathogen. Once the disease-causing pathogen is killed, these antibodies stay inside the human body itself, so that when the individual is coming across the same pathogen for the next time, it reacts with the pathogen quickly and prevents any further infection. Based on the same principle, vaccines are produced, and these vaccines consist of dead or weakened disease-causing microbes, and once they are injected into the body, they produce antibodies. And the antibody which is produced remains in the body and protects it from any future attack of disease causing germs, and henceforth the body is said to be immunized. And since vaccines are increasing the immunity of an individual, vaccination is also called as immunization, and vaccination is a process of giving vaccine to the individual, which can be administered either through oral drops or through injections. Vaccine for smallpox. It was in 1798 an English country doctor, Edward Jenner, who discovered the vaccine for smallpox. And Edward Jenner was an English country doctor, and he administered the world's first vaccination as a preventive treatment for smallpox, a disease that had killed millions of people over the centuries. Edward Jenner noticed that milkmaids who had contracted a disease called cowpox, which caused blisters on cow's udder, did not catch smallpox. Unlike smallpox, which caused severe skin eruptions and dangerous fever in humans, cowpox led to few ill symptoms in these women. So, he took a fluid from cowpox blister and scratched it into the skin of a boy called James Pepys, an 8-year-old boy. A single blister rose upon on the spot, but James soon recovered. On July 1st, Jenner inoculated the boy again, but this time with a smallpox matter, and he observed no disease was developed, and the vaccine was a successful one, and doctors all over Europe soon adopted Jenner's innovative technique, leading to a drastic decline in new sufferers of the devastating disease. Harmful microbes. Pathogens. So what are pathogens? The disease causing microbes. Those microbes which are going to cause diseases in human, animal and plants are generally coined by the term pathogens. And these pathogens can get transmitted by direct contact with an infected person or a carrier by an animal. These microbial diseases, which can spread from an infected person to a healthy person through the different means like air, water, food and physical contact, are generally called as communicable diseases. And examples of such communicable diseases includes cholera, common cold, chickenpox and tuberculosis. So, what are communicable diseases? The microbial diseases that can spread from an infected person to a healthy person through air, water, food or physical contact are called communicable diseases. For example, when a person suffering from common cold sneezes, the fine droplets of moisture carrying thousands of viruses are spread in air, and these viruses may then enter the body of a healthy person while breathing and cause the infection. And apart from this, there is also one more method how a communicable disease can spread from one person to another. That is through vectors or carriers. There are some insects and animals which act as a carrier of disease-causing microbes. Housefly is one such carrier. The fly sits on the garbage and animal extra, and pathogen sticks to their body. When these flies sit on uncovered food, they may transfer the pathogens on the food. Whoever eats the contaminated food is likely to get sick. So it is always advisable to keep the food covered and avoid consuming uncovered items of food. Another example of carrier is the female Anopheles mosquito. And this female Anopheles mosquito carries the parasite of malaria, that is Plasmodium, and female Anopheles mosquito acts as a carrier of even dengue virus. And how can we control the spread of this mosquito? Because always these mosquitoes breed in water. So make sure that you do not let water to collect anywhere in coolers, tires, flower pot nearby your surroundings. So by keeping the surrounding clean and dry, we can prevent the mosquito from breeding. And how do this spreads the disease? When it is going to bite a infected individual, the virus enters into the body of the insect, and now when it is going to bite a healthy individual, it is going to inoculate the virus into the body of the healthy person, and finally now the healthy person is also infected with the virus. Now let us discuss about few common diseases caused by microbes in human. The first is tuberculosis, and tuberculosis is a bacterial disease and it is transmitted through air, and measles, chickenpox and polio all are viral diseases and they can be transmitted either through air or through physical contact or by water. And what are the general preventive measures? You can keep the patient in complete isolation and make sure that the personal belonging of the patient is away from those of the others, and vaccination should be given at the suitable age in order to prevent the occurrence of these diseases. Coming to cholera and typhoid, both are bacterial diseases and the mode of transmission is either through water or food. It is always made good to maintain personal hygiene and good sanitary habits and consume properly cooked food and boiled drinking water, and always go for vaccination in order to protect you from the occurrence of these diseases. Hepatitis B, and it is a viral disease and it spreads through water, and the way we can prevent is by drinking boiled water, and also we should take vaccination at the proper timing. Malaria, it is a protozoan disease because it is caused by a protozoa called Plasmodium, and it is a vector transmitted disease because it is being transmitted from the infected person to a healthy person through the mosquito. The general way how we can prevent is use mosquito net and repellents, and also spray insecticides and control the breeding of mosquitoes by not allowing water to collect in the surroundings, and protect yourself from the mosquito bites. Now let us discuss about few diseases caused by microbes in plants. The plant disease citrus canker, it is caused by bacteria, and how it spreads? It spreads through air. And rush of wheat is also caused by a fungi, which is a microbe, and it also spreads through air as well as through infected seeds. And yellow vein mosaic of bindi, that is lady's finger or okra. It is a viral disease which spreads from the infected plant to a healthy plant through the carrier insect. So, what does the word food poisoning means? Any undesirable change in color, odor and taste in the food brought about by microorganism is called as food poisoning, and this food poisoning may cause infections, serious illness, and even at times it may lead even to the death of the individual, and food has to be preserved in order to prevent it from being contaminated by microorganisms. What is the characteristic of a spoiled food? The food which is contaminated by bacteria or any other microorganism is unfit for consumption. That is, we should not eat those contaminated food. And these contaminated food are only called as spoiled food. And we can easily identify them by change in color and stickiness and bad odor and bad taste, and even the consistency will vary. In order to prevent the spoilage of food, we have to preserve the food. And there are several methods of preserving the food. And they are chemical methods, preservation by vinegar, preservation by sugar, preservation by oil, sun drying, salting, pasteurization, boiling and freezing. And what does the chemical method of preservation means? It is a method in which certain chemicals are being added so that the food will not be spoiled, because these chemicals will inhibit the growth of the microbes. And in case of preservation by vinegar, vinegar is being added to the food items, and on adding vinegar, the sourness of the food increases and the microbes cannot survive in high sour environment. And preservation by sugar. By adding sugar, the moisture content of the food is reduced, and moisture is one of the important requirements for the microbes to multiply or proliferate. And once the moisture content is removed from the food, then the food becomes something which is not suitable for the growth of microbes. Preservation by oil. On adding oil to the foodstuffs, the growth of bacteria and fungi can be inhibited, because fungi and bacteria cannot grow in the environment which is rich in oil. Sun drying. Sun drying is a method where the fruits and vegetables are made into thin slices and then they are dried under sun, so that the moisture will be completely dried off and the microbes cannot grow anymore in those food items. Salting is a procedure where excess amount of salt is added to the food. On adding salt, the water content from the food is being absorbed, and finally the food becomes dry, and in any dry food substance the microbes cannot grow, and hence the food is being preserved and protected from the growth of microbes. Pasteurization is a method of preserving milk, and it was first demonstrated by Louis Pasteur. And during pasteurization, milk is first heated about 70 degrees Celsius for 15 to 30 seconds and then suddenly chilled. And this sudden chilling of milk is done to prevent the growth of microbes. And boiling and freezing. Boiling and canning is a method of preserving drinks, colas, beverages and other products which are to be stored under low temperatures, and carbon dioxide is added as a preservative in some cold drinks. Some edible foods like fish, meat are canned in airtight containers for preservation. Nitrogen fixation. Our atmosphere contains a lot of nitrogen gas. In fact, our atmosphere has 78% nitrogen gas. The atmospheric nitrogen gas cannot be utilized directly by plants or animals. In order that nitrogen gas of atmosphere can be utilized by plants for their growth, but it has to be first converted into nitrogen compounds that can be absorbed by the roots of the plant. And the process of converting this nitrogen gas of atmosphere into compounds of nitrogen which can be used by plants is called nitrogen fixation. And for this purpose, there are certain bacteria which are exclusively meant for this purpose, like rhizobium bacteria, blue-green algae, etc., which lives in the root nodules of plants. And this process is done exclusively by a part of nitrogen cycle. So now let us discuss about nitrogen cycle in detail. Nitrogen is required by both plants and animals for their growth and development. And nitrogen is an essential component of protein which make up the body of plants. And nitrogen is also present in chlorophyll, nucleic acids and vitamins. The same nitrogen element is circulated again and again through living things like plant and animals and non-living things like air, soil and water. The circulation of nitrogen element through living and non-living environment is called nitrogen cycle. So, the atmosphere contains nitrogen gas and the nitrogen fixing bacteria present in the soil, in the root nodule of leguminous plants like blue-green algae and lightning in the sky fixes nitrogen gas from the atmosphere and convert it into compounds of nitrogen which goes into the soil. And the second step is the plants take the compounds of nitrogen from the soil for their growth, and the plant absorb the nitrogen compound from the soil through their roots, and the plants convert the compounds of nitrogen into plant protein and other organic compounds which make up the body of plants. And the plants are eaten up by animals as food, and animals convert plant protein into animal protein and other organic compounds which make up their body. And some animals also eat other animals to obtain nitrogen compound. Thus, animals obtain nitrogen compound by eating plants as well as other animals. When plants and animals die, the complex nitrogen compounds like protein, etc., present in the dead body of decomposed and are converted into simple compounds of nitrogen by certain bacteria and fungi present in the soil. And animal excretions are also converted into simple compounds of nitrogen by bacteria and fungi. All the simple compound of nitrogen formed in this way goes where? It goes into the soil. And the compounds of nitrogen which were taken up by the plants from the soil during their growth are now returned to the soil, and from the soil these nitrogen compounds are again absorbed by new plants for their growth, and this part of nitrogen cycle repeats endlessly, whereas some of the compound of nitrogen formed from the decay of dead plants and animals are decomposed by denitrifying bacteria which are present in the soil to form nitrogen gas, and this nitrogen gas goes back in the atmosphere from where it initially came, and this process is the reverse of fixation of nitrogen. In this way, the nitrogen gas which was removed from the atmosphere during fixation is put back into the atmosphere. From the atmosphere, nitrogen gas is used again during nitrogen fixation, and the nitrogen cycle is repeated in nature again and again. As a result of nitrogen cycle in nature, the percentage of nitrogen gas in atmosphere or air remains constant. So, the nitrogen gas from the atmosphere is fixed into the soil, which is called nitrogen fixation, and from the soil it is being absorbed by the plants and used for preparing amino acids which are then used for making up proteins, and from the plants it is reaching the animals, and then Once the plant and animals are dead, these complex nitrogen compounds are broken down into simple compounds, and again it is mixing up with the soil from where it reaches the plants once again. Whereas during this decomposition process, certain amount of nitrogen is released in the form of gas by denitrifying bacteria, and the process is called denitrification, which is the opposite of nitrification, and it is released back into the atmosphere, and this entire cycle is repeating endlessly or continuously, as a result of which the percentage of nitrogen in the atmosphere or in air remains constant. Thank you, students.